Hello, I'm María Isabel Gómez. I'm a research associate in the Cassava program at CIAT based in Colombia. Today, in this presentation, I want to give an update on the results of diagnostic and surveillance of Cassava whiteflies activities in mainland Southeast Asia. The whiteflies of the complex Bemisia tabaci are the most important vectors of viruses. In the last years in Cambodia and Vietnam, outbreaks of CMD caused by Sri Lankan Cassava mosaic virus were found to be associated with Asia-2-1. This is a whitefly from this complex and also is the only known efficient vector for this virus. So we established a method fast, efficient, and standardized for the surveillance of whiteflies and diseases. For whiteflies, we evaluated 30 plants following a diagonal in each of 1,335 fields. The protocol was a result of the interaction with the partners from Laos, Cambodia, and Vietnam, and they carry out the samplings in the field, collecting adult whiteflies and photographs of different parts of the plants. For whiteflies, we established to evaluate the second youngest leaf based on the preference of whiteflies in Latin America and some experiences in Uganda. It's important to highlight that the species that are found in Southeast Asia are different than those that we evaluate in Latin America and in Africa. The variable we used to measure the populations was the relative abundance, which is the number of adults per second leaf per plant. In the two consecutive crop cycles, we obtained relative abundances of 0.068 in 2020 and 0.267 in 2021. The photo records allow us the recognition and counting of 2,800 whiteflies. More than 22,400 photographs were obtained, and from those, 15,000 was from the cycle of 2020 and 6,600 were obtained in 2021. We used the data from the two crop cycles to visualize the abundances in the region. In the map at the right, each circle contains several fields, and the number in the center is the average relative abundance of those fields. The data from the 2 years were organized in 6 ranges, which were established according to the number of the fields in each one. The first range in dark green corresponds to the 394 fields where no whiteflies were found in the photographs. Few fields had more than 0.5 whiteflies per leaf, just 22 from 750. In Cambodia and Laos, the abundance was lower in 2021 than in 2020, while in Vietnam the average abundance increased from 0.035 to 0.345. The number of fields was lower in 2021 because different reasons: change of crop in the field, COVID restrictions, etc. When look at the north zone of the region, we found that most of the fields have less than 0.04 whiteflies per leaf. However, at some fields, the populations are a little higher, and for instance here, close to the province that has some cases of CMD, there are a small population, a little more higher, of whiteflies. The middle zone, which includes the borders between the 3 countries, more fields have more than 0.04 whiteflies. This is a lower number compared with the records that we can find in other parts of the world, like Latin America or Africa. But into the region, these are intermediate levels of population. Interestingly, in this zone, there are several fields with CMD. At the south, not much fields have hive population, but in the zone we found the 16 fields with abundances between 0.5 and 1, and also the 6 fields with an abundance between 1.1 and 20 whiteflies per leaf. As a reference, a population of 100 whiteflies per 5 leaves was considered as superabundant in Africa. This zone also have most of the fields affected by CMD. Although in a wide vision of the maps we can see any relation between the incidence of CMD and whiteflies in some regions, we didn't find a correlation between these two variables. Low and high population of whiteflies were found in crops with low and severe incidence of CMD. However, in all 6 fields with the highest populations of whiteflies, the fields have an incidence over 98.33%. The data could suggest that the contribution to the CMD dissemination is mainly by stakes, and when the field has high incidence, the populations of whiteflies can increase. In the graphic of the lower right, we remove the outliers to see the distribution of the points of the abundance and incidence. So it is possible to see that the surveillance protocol allowed to map low and high populations of whiteflies in the region. However, every protocol can be improved, and we wanted to evaluate if the preference of the whiteflies for the different leaves was the same as that found in other conditions. 17 plants were evaluated in Vietnam, and we found that the preferred leaf is the fourth, not the second, for that conditions. This is a result that we need to confirm with more samples. To know how much the whitefly transmission can be increased in the dissemination of CMD, we can use the photos from previous cycles for surveillance of Cassava, which is broom, to evaluate the contribution of whiteflies in the dissemination of the disease, checking in what level of the plant started the symptoms. If the symptoms are since the base of the plant, as in the photos, the stake was infected by the disease from the beginning. In each field, together with the photographs, were collected adults for identification. The koi barcoding protocol were implemented, and samples from the cycle of 2020 from Laos were analyzed using nanopore in Colombia, and samples from Vietnam were analyzed in PPRI. In both cases, the cryptic species Asia 2.1 and Asia 2.6 were identified. Given delays in transport samples due to COVID restrictions, we have pending to analyze the diversity in samples from Cambodia. It is relevant that in both cases, Laos and Cambodia, one of the cryptic species collected was Asia 2.1 because, as I mentioned at the beginning of the presentation, this is the most efficient vector of the Sri Lankan Cassava mosaic virus. So we use the known distribution from our data and literature records of this species to generate a potential distribution map. Given in some regions the population of whiteflies is increasing, it is required to take management practices, as the use of varieties resistant to CMD but also to whiteflies to control the increasing populations. To use the high-throughput phenotyping platform can be useful to evaluate the resistance to whiteflies of Southeast Asia varieties. This is a platform developed by CIAT. Finally, to summarize the main points of the presentation, the surveillance protocol allowed to map low and high populations. However, we are required to evaluate the preferred leaves of the whiteflies in the region. It isn't required to evaluate alternative hosts for the whiteflies, given that Asia 2.1 uses at least 11 species as hosts. The contribution of whitefly transmission to the dissemination of CMD can be evaluated from the Cassava, which is the broom disease photos from the cycles of 2020 and 2021. Also, it's possible to evaluate Cassava bacterial blight from these photos, as well for the surveillance activities that will be made at the 2022 cycle. It is pending to analyze the diversity of whiteflies from Cambodia. Thank you for your attention.